So biological control starts with natural control, what's actually going on in the environment. So in this week's lab, what I'm going to be doing is taking you around and showing you what kinds of hosts and natural enemies are happening in basically a native environment. So I'm in Polly Canyon, and this area is, basically just as is typical riparian chaparral type habitat, and we've got cottonwoods, we've got sycamores, we've got oaks, willows, and those would be the tree situation. We've got different kinds of shrubs like coffeeberry and baccharis, the California sage, artemisia. We've got a variety of different things. So I'm going to take you around and show you those things. And what I want to get across to you is that biological control basically has to start in a setting like this. And when we talk about a pest that's introduced, that has been moved out of its area of origin into a new area and become a pest there, this is the area of origin, something like this. So this is where we need to start and understand that everything here is in balance. And we've talked about that before in APS 313. So if we had an organism, an herbivore that was moved from its native area and became a pest, and we were going to do the biological control method of introduction, we'd send a foreign explorer to that area and they would start looking around for that organism. And in the native area, of course, it's going to be in low numbers because it has lots of other species that are helping to keep it in check. All the mortality factors are in place. Whether that's the competition that I was talking about, natural enemies, diseases, all kinds of things are going on to keep everything in balance. And that's what you see basically here in Polly Canyon, a lot of examples of that. Now, you might look at this landscape behind me and think that it's desolate and that there would be nothing here to examine, but in fact there is a lot going on here. And that's the crucial thing about biological control, is I've got to get you thinking on a much smaller scale than you're used to thinking. Okay, and so I want to point out this plant right here. This is called tarweed, and when you take it and pick a little flower off, it is extremely odoriferous, and the odor that it reminds me of, I would say, is like horse pee pee. It's a nice way to put it. And it's a little composite. So it's got the ray flowers, and inside there is the seeds, or the achenes, and there is a taphritid fruit fly that's a native. It doesn't feed on fruit, it actually feeds on these seeds, and it will lay its eggs in this flower head. The larvae then will hatch out, and then those larvae get attacked by parasitoids. And so when you look at something like this, you think, what in the world could be going on? This looks desolate, there's nothing, it's just, empty. Oh, no. So there's a fly that uses this as a resource, and then that fly has natural enemies. And so one of the things that you can do is come along and collect these flower heads, put them into a container, like I showed you last week, and then wait for the magic to happen. And if you have a bunch of these flower heads in a container, what you're going to see are the flies emerging and their parasitoids emerging, and then you're going to have that association of natural enemy and host. So oaks in California provide a really interesting resource for this class, for looking at hosts and natural enemies. And one of the one of the ones that I kind of focus in on with the class are the galls that are on the oaks. And here's one here. Now, this gall is, you can see it's brown, it's hardened. This is now basically a fully mature gall. And you don't see any holes on it, so whatever made the gall is still inside. Now, the gall is made by a gall wasp, a cynipid, and you have seen that process in the video, the first lecture for this week. And so back in the spring, there was a gall wasp that laid her eggs on that stem that created the place where the eggs would hatch out and the larvae would feed. And their feeding, as well as their saliva and their cuticular hydrocarbons, then set the stage for this chemical and physical interaction that results in a very specific looking gall. Now this one looks different from that one in the video, doesn't it? That's because this is a different species, and you can tell what species that's in the gall by the shape of the gall. All right. Now, this gall will also be a magnet, as it's developing, will be a magnet for parasitoids. And some of the parasitoids, the burglar that David Attenborough referred to, will have long ovipositors, those egg-laying tubes, and they will drill in and find those grubs inside that are feeding and creating this gall, and then lay their eggs in there. So instead of getting a gall wasp chewing its way out and creating a round exit hole and then leaving and completing its life cycle and starting the cycle all over again, you will also get these parasitoid wasps chewing their way out, making an exit hole, and then they're going on their own life cycle, mating and looking for galls to lay eggs into. So oaks have a tremendous number of galls, a lot of different species. This one is a leaf fold gall. So there you can see the leaf is folded over and there's a little pustule there. This is one that's kind of dyed. There's some more over here, and then this one here. Now this is made by a fly. The fly is in the family Cecidomyiidae, which are the gall midges. Now you're going to have to get to know that name, Cecidomyiidae, because not only are there gall midges, that kind of interesting biology, but there are predatory species that are also commercially available that we use for aphid control in biological control. So these guys, what I'm showing you is opportunities for you to go out and do some collecting. If you can find a live oak like this one, or some of the deciduous oaks like we have up in North County, they're going to be loaded up with all kinds of galls. And if you can get out and find some of these and then put these away in those cups like I showed you, you're going to be able to get out both the hosts and the natural enemies, and you're going to see this association. This one I wanted to point out because it's kind of interesting. The gall is known, and but no one has really ever had a successful gall maker emerge. We don't know what they look like, nor do we know if there's any parasitoids associated with them. Obviously there should be. So maybe you'll be one of the lucky ones that collects one of these galls and has something emerge, and you'll be able to help us figure out this scientific mystery. All right, well, another star of the video that you saw with David Attenborough was the California oaks that had the little pearl galls on the leaves. And remember how the pearl galls would pop off and jump around? All right, well, this is a close relative. That one in the video lives in Southern California. This is the one that we have here in San Luis Obispo. Now, you saw the size of that gall, right? It's very tiny. And you may be asking yourself, oh, is there actually really anything in there? And there is. There is a tiny little gall wasp that's in there that also gets parasitized by a little tiny gall wasp parasitoid. And that's why I was telling you earlier, we've got to get your focus down to this microscopic scale. in order for you to fully appreciate all of the natural control and biological control that is happening every day right under your nose. And this giant swelling on this stem is yet another kind of gall wasp. And this one is, obviously there's no exit holes, and it is in peak ripeness, if you can refer to galls being ripe. And we should be expecting to see some really incredible gall wasps and their parasitoids emerging from this within a few weeks. So, in lecture this week, I'm talking about ecological succession. And remember, that's the secondary succession, where we're going from disturbed habitat to pioneer species of plants that are settling that area down, and then moving into more perennials, stabilizing the community, and then eventually going to, a climax community. Well, here's a really good example. We've got this area here that's full of all these different grasses, and this would have been a disturbed area, and it's got the grasses, and you can see some of the turpentine weed or tar weed, and you can see some Nephalium, the pearly everlasting. And so what we've got are some plants that now are starting to come in and stabilize post pioneer species, and this eventually will be taken over by species that you see here, like Baccharis. This is Baccharis, coyote brush, and this is a crucial plant in California's native systems. And you may have seen it just along the roadsides. It's a plant that can handle disturbed habitats, very unforgiving soils, and so forth. Does great. It plays such a crucial role. So there's a couple of things I want to kind of talk about. One is its natural native role, and then the other is the role that it plays as a component of conservation biological control. So the natural thing, the native thing that it's doing is that this is considered to be a stabilizer for an area like this, but also an oak tree nursery. And the reason that this is considered an oak tree nursery is because that our scrub jays that we have will go to the climax community oak trees that you see over there. They will get those acorns, and then they'll start poking them into the ground, and they'll kind of just put them everywhere, but they'll put them up underneath plants like this Baccharis. Now, in their minds, they're going to come back and find those acorns and eat them later, but they forget. And so what happens is that those acorns are left, germinate, and then the tree that's starting to develop is protected from browsing from deer, because this kind of keeps it under cover. Eventually, the tree grows up, gets beyond this, and is okay, gets kind of beyond the susceptibility to being fed on by deer. And then we'll see this kind of a community where it's mixed, giving rise to that next community there, the climax community. The role that this plays in conservation biological control is that it's considered an insectary plant, a hedgerow plant. And I'm going to be giving you resources about that later on in the quarter. And these flowers here on this plant are the female flowers. And so Baccharis is considered to have two houses. I forget, is it monoecious or dioecious? All you plant people can tell me later. Email me. I don't know. But I know that this is a female plant and this is a male plant over here. Different shaped flowers. Now, these flowers both are providing resources for parasitic wasps, predatory wasps, flies, and other pollinators. So they are coming here and getting nectar, getting pollen. There's also plant resins that are associated with this. And these plants also harbor sternorophorans that are producing honeydew. And so they're on those and feeding on the honeydew. They're feeding on all kinds of stuff. It provides shelter and food because those female wasps that are out looking for hosts, they need fuel too. And this is where they gas up and they rest up. And so this is a crucial plant that provides that shelter, and that's why it's so particularly good in hedgerows on farming systems where you're trying to enhance biological control, native biological control, because it's so easy to grow and maintain. You don't even have to worry about it at all. There's going to be a video later on that's going to be talking about a hedgerow planting that's in Salinas at the USDA organic farm there, and how that came to be and what it looks like today. And this is a major player. So here is a gall that's on Baccharis. How about that? Now, you can collect these galls, and you can collect the flower heads, both the males and the females, and you can put them away for your project. But you got to get a visual on these galls. So you may not have seen it, but there it is. And then look, here's one here. Look at that right there. And then here's one here. What? Check that out. All right, now this one, that's a big one. So pretty. Now this one, I'm gonna say, this is the first one we were looking at, and I would say, oh yeah, collect that. But then I turn it over and I see a hole. That's the exit hole. And that means whatever was in there has now emerged. And so don't collect that one because you won't get anything out of it. So this plant here is Artemisia californica, California sage. And you can see these little balls hanging there. Those are the flower heads. And it's got a great odor. That is, unlike the tarweed, this is just lovely. And indigenous folks use this as a cure for headaches. Shove it up their nose and get rid of the headaches. You cannot do that. One of the things that I want you to do, though, is be careful if you're out like this and you're following my lead. Watch out for snakes. So stomp around, wake up the rattlesnakes, make sure they're not there. And also watch out for ticks. Okay, so I got to do all this, you know, whatever. It's the age of COVID. Look out for everything. But you can collect these flower heads. You can just basically take these little stems and strip them of their flower heads. Put a few of those in those cups, and you will get out the herbivores that are feeding in that flower head and their parasitoids. This is willow. There's a couple of different, I guess, species. This one is the rounded leaf, and there's another one that's got a pointed leaf. And they have a lot of activity going on, and galls is one of them. So what you see here is a rosette gall, and there is a gall maker in there somewhere. And so you would collect, just cut this off, put it in a container, get rid of a lot of the extraneous leaves, and down in there somewhere is a gall maker. And the base right down in here. So what you'd do is you'd come along and just cut it off right here, get the excess leaf material off before putting it in a container, and then just let it do its thing. Now there's ants all over this, and when I pull that leaf away, you can see that there's some white stuff in there. I'm guessing that's mealybug. And the ants, of course, these are Argentine ants. They are after the honeydew and protecting those guys, so there might be an added bonus. But let's focus again on, get back focusing on the galls. So here's some other type of little bud galls that you can see on willows. And they're smaller than that one that was over there that I just showed you. But these will have gall makers and parasitoids in them, so don't hesitate. There's one under there. Check it out. And here we have a leaf gall, and these occur on the veins of the leaves, and they can be either on the under or upper side of the leaf, and they typically turn red this time of year. So what you would do is collect the leaf and then go ahead and put that into a container and wait for whoever's inside of there to make their way out. Now, it may take a few weeks, maybe even a month, and in some years when we had the class and we were doing the collection, we'd be doing these collections early in the quarter, like the first 2 weeks. You got to get this done because it takes a while to get all this stuff out and emerged and then identify it and curate it and turn it in for the collection. So you guys are going to be kind of on the same timeline. So I want you to get out and explore, check things out, and collect. Just collect things and get them into your containers. And if you're going for the galls, you can go out any time of day, it doesn't matter. But if you're going to be looking for those predators, well, make sure that you get to an area like Polly Canyon, something like that, where there's a lot of stuff in bloom, even in landscapes, in parks and other things, where there's lots of flowers in bloom. Go between the hours of 10 and 2. 10 a.m. to 2 p.m. because that's going to maximize your chances of finding those predators out and about so that you can see them. This giant thing is a scale, and it occurs on oaks. And guess what? You can see the little hole there? The parasitoid's already emerged. If you happen to find some of these on a live oak and there is no hole, go ahead and collect them. And obviously you will get a parasitoid out, especially if in your area it isn't quite as far along as it is here. There's another one up here. And maybe you'll get lucky and see what the parasitoid is that comes out of that.